as you all are familiar, usually we have Thursdays as a seminar, not only for scholars but also for our eminent scientists from within and even from our partner institutions. So it's a pleasure that two of our scholars finishing soon, and one of the requirements is, of course, the presentation of their PhD thesis. So for this morning, MSc, yeah, okay, MSc, I mean MSc, when I say scholars, MSc and PhD. Okay, so that's one of the requirements. So for this morning, we're having two seminars. One is from Uttam Chan, who will be discussing genotype and environment interaction in pigeonpea hybrids Cajanus Cajan. And the other one, of course, let me also introduce at this point, you can come one after the other. Sunil Chowdhury, who will be presenting stability of maintenance of male sterility and fertility restoration in pigeonpea, also Cajanus Cajan, and their eminent supervisor, scientist supervisor at ICRISAT is no other than Dr. KB Saxena, who is the father of pigeonpea hybrids here. Yeah. No, sincerely, who has really started and did much work, and of course the success of the hybrids production here in the world, and of course here in India. So without much ado, let me call on Uttam Chan to do his presentation and to be followed by Mr. Chowdhury. Thank you and a pleasant morning again to all.
Good morning, all of you, and thank you, madam, for giving the introduction. And myself, Uttam Chan. Today, I am presenting my thesis seminar. My topic is genotype cross environmental interaction studies in hybrids and light cultivar of pigeonpea. In my seminar, I will cover these topics. First of all, I am talking about the introduction. As you well know, pigeonpea is the often cross-pollinated crop, and India is the native place of the pigeonpea, according to vendor Mason. Okay, sir. Thank you, sir. And pigeonpea genome size is 858 mega base pair. It's pigeonpea have the 18 to 24%, and peculiar feature of the pigeonpea, it has the only pulse crop having the hybrids at commercial level. Now I am telling about the pigeonpea production status of the India. It is the second most important pulse crop of the India, mostly used as dal, and here you can see red bubbles are there. They are the major growing state of the pigeonpea in India. Bihar, Madhya Pradesh, Maharashtra, Andhra Pradesh, Karnataka, Jharkhand also, and that is the productivity of the pigeonpea at global level and Indian level. Now, area production and productivity, you can see the graph of 10 years, more than 10 years, 2000 to 2011-12. In this graph, you can see the productivity not increasing, but area and production increasing every year. Why area of pigeonpea is increasing? Because pigeonpea is previously the long duration crop. Because now in pigeonpea, so many research and so many other things. Now early varieties are available, like ICPL 88039. So farmers are ready to grow the pigeonpea in our on field, and that's why pigeonpea area is increasing in so many areas of the country and that globally. Now I am coming to my topic. Importance of G into E interaction. What is the importance? Why are you doing this thing G into interaction? Bioadaptation, consistent performance of released varieties. We identify the stable genotype in different environment or not. Yield stability performance across varied environment, and phenotypic response change in environment is same and different for all the genotype or objective. Now coming to my study objectives. First objective is to determine the environmental influence on yield and yield characters. In second objective I covered to determine the flexibility in genotype to the environment for stability and yield characters. Third important objective to identify the hybrids and cultivar for stability, desirable yield and yield components. And fourth, to identify the stable pigeon pea hybrids which can be grown in wide range of environments. These are the objectives. Now coming to my experimental material. So first I want to tell my material developed by the crossing A line and R line. So I use 5 A lines and 6 R lines, fertility restorative line. I used 12 genotype including 10 hybrids and 2 checks, 2 standard checks, Maruti and Asa. Detail of 3 environments. I used multi-location trial for the study of G into interaction, ICRISAT Pathancheru, BAU Birsa Agriculture University Ranchi Jharkhand, and RAK College Sior MP. And this is the latitude, different latitude, longitude, altitude, soil type, different climatic zone. And one more peculiar thing in my study, the different date of sowing and different date of harvesting. That is the important phenomena of the multi-location trial of my study. Observation recorded. I recorded these observations for my study, and on the basis of these observations, I find out my result. Experimental detail. Already I told I used 12 genotypes, 3 locations, and I used randomized complete block designs, and genotypes grow in 2 replications, row length, spacing, row to row spacing, number of rows. Data collection. Genotypes were grown randomized complete block design with 2 replications at 3 different locations, and data were collected from 5 randomly selected plants for different characters. Data analysis. Data obtained from multi-location trial and analysis by the GenState software from our biometrical unit. Stability analysis. This is the important. In stability analysis, I used 2 methods. First one regression method and second one GGY plot method. First of all, I want to tell about Everett-Russell model. Everett-Russell model is total variance first divided into two components, genotype and environment plus G into E interaction. And after that, second component is environment plus G into E interaction, further subdivided in 3 components, environment linear, G into E linear, and pool deviation. And pool deviation are further divided in sum of square due to individual genotypes. And this model consists of 3 parameters: mean, regression coefficient, and deviation from the regression line. And this is the criteria, this is the parameter for the stable genotype. How to say this is the stable genotype or not? So Everett-Russell and given the 3 parameters, mean should be high for the stability of any genotype, regression coefficient should be near to unity, and deviation from the regression should be minimum. That is the criteria for the stability of. And I used second method. This is the GGY plot. This method given by Yan et al. And this is the latest and very effective method. And in this method, we are the GGY plot method is the graphical representation of the data and of the genotypes environment. And why GGY plot method is more effective? Because it will give the information mean performance of genotype and environment also, which environment is good or which environment not good. That is the main difference between GGY plot and Everett-Russell model. That's why mainly today we are following mainly GGY plot. steps in GGY plot. How to interpret the data, how to came result from a GGY plot. First of all, we arrange the data and centering the data. After that, in parting single value into genotypes and environment score for each principal components. We are given PC1, PC2. For stability of the ideal genotype, PC1 should be high and PC2 should be less. PC1 high means is the mean performance of the genotype, and PC2 low. It means it is the stability of the genotype. Result and discuss. We are coming to result. Pooled analysis of variance. In this you can see all the genotype highly significant. Genotypes also highly significant for these characters. Environment also significant. G into E interaction also significant for each and every character. So we can go for a stability analysis for these characters. This is the fulfillment the criteria of the stability analysis. And this is the continue for another characters. Only for we can see G into E interaction, 100 seed weight is coming non-significant. All other characters also showing significantly. Only one character are there, G into E, not showing significantly. This is the another table given by regression method. In this table, you can see genotypes are highly significant, expect days to 50% flowering. Environment G into E interaction are also non-significant for maximum characters. Environment linear is highly significant, except days to 50% flowering. G into E linear is also highly significant, expect days to 75% maturity. G into E linear, environment linear also how to come significant? They are tested by the pool deviation. Continue remaining characters. In remaining characters also G into E interaction for 100 seed weight also coming again here non-significant. Now we are going for stability parameter. After coming to G into E interaction is to non-significant, that's why we are going for stability analysis. And three parameters I told three parameters. Mean, regression coefficient, standard deviation. In that for days to 50% flowering, 3933, 2671, 3494 are the stable. But 3477 non-stable, not stable. We can't say this is stable because the regression coefficient is so it's not in linear fashion. All the regression coefficient are in linear fashion, but is not near to unity and not in linear fashion. According to this model, regression coefficient should be linear. That's why you can't say 3477 is stable. And for days to 75% maturity, maximum genotype are the stable. But in plant height, for plant height you can see the red circle, the genotype and this one too, they are the stable genotypes. And other genotypes are non-stable compared to check and other things. For stability parameter for primary branch, secondary branch, spots per plant, you can see 2671, 4490 are stable for primary branches. Why they are stable? Because high mean and regression coefficient near to unity, and standard deviation not near to 0, but this is two criteria are the fulfillment by the genotype. That's why we can say this is the stable genotype. For secondary branch, 2740, 4490, 3494 and 3491 are the stable. For power per plant, it is a very important character for yield and yield contributing characters, powers per plant. So we can say 3933, 2671, 4490 and 3762, 3461 are the stable for powers per plant. Stability parameter for seeds per grain. Now we are coming to yield and grain yield and kg per hectare, grain yield per plant. But here some deviation is coming because grain yield kg per hectare estimated only based on plot yield, and yield per plant is different. That's why some deviation is coming here. 4490 is stable in grain yield kg per hectare, but grain yield per plant is not stable. And 3762 is stable, here also stable, but 3491 not stable for plant yield, grain yield per plant, but 3491 is stable for grain yield per plant. So grain yield per kg per hectare estimated based on plot yield, that's why this variation is coming. Already I discussed GGY plot methodology, so now I am telling about the GGY plot, how to result is coming from GGY plot method. Here two axes are there. This is AE axis and this is AEC axis, average environmental coordinate axis, and this is AEA, average environmental axis. So we can say what is the mean performance from this axis of the genotype. We can say in this circle, we can say in this circle this is the stable genotype. Lowest distance from the AE axis, this is the define the stable genotype. And and we are seeing the location. Simultaneously we can see the location. In location, Ranchi is the closest to concentric circle. Small circle is there. Ranchi close to concentric circle. So we can say Ranchi is the best location for days to 50% flowering. According to this figure, and Sior and Patancheru are so many far, they are not good for days to 50% flowering, means coming early flowering in Ranchi location, and Patancheru and Sior coming some late flowering according to this graph. And days to maturity, here we can see Ranchi also days to Ranchi also coming so many far from this circle, so we can say this is not good for days to maturity. Here and Ecrisat and Sior. Ecrisat, if we will see the angle like that, so Ecrisat is so far, Sior also so far. So we can say Ranchi, Sior, there is no correlation between the three locations at the days to maturity. And genotype 6 is the best location in Ecrisat, and genotype 5 the best location in Sior. But overall we can't say this is the stable genotype for all three locations, because the mean performance from the AE axis is more in 6 genotype in Ecrisat and 5 in Sior location. So we can say this is the not stable genotype for days to maturity. Coming for next plant height. In plant height, Ecrisat is the best location, ideal location, because it is coming on the axis and near to axis. And genotype 4, 5, 9, 6 are the having low mean performance from the AE axis. That's why low distance is there. That's why we can say this is the stable genotype for three locations. And genotype 10, genotype 8 are genotype 10 is best in Sior location and genotype 8 best in Ranchi location. So we can say based on the distance from the AE axis and near to concentric circle, we can say four genotype is best in overall three locations. So GGY plot also gives the idea about the location plus genotype, and Everett-Russell model given on the idea about the genotype. For same thing in for primary branches. Primary branches genotype 7 and genotype 2 is best in Patancheru location, but not in the overall two locations. Here Sior and Ranchi is coming downside of the AE axis and Patancheru coming over the axis. So we can say genotype 6 and 1 is stable in three locations, but not in the three overall, no 7 and 2 not in the stable in three locations. 7 and 2 are very good in Patancheru location. Coming to pots per plant, here you can see Ranchi, Sior, Ecrisat. Ecrisat so many far. If we will say the angle from this cross point to Ecrisat, Sior, Ranchi, if we which location angle is low, that is the good location for this character. And we can see the Ranchi location make the lowest angle, after that Sior, after that Ecrisat. And so we can say Ranchi is the best location for pots per plant, and genotype 2 is going showing the stability from this axis to overall three locations, and genotype 9 and 3 is the best genotype in the Ecrisat. Grain yield per plant, Ecrisat and Sior location, you can see they are both are correlated, and both are showing for this character same performance for genotype, and Ranchi is variated from that. And you can see the genotype 5 is the so much distance from the AE axis, so we can say 5 genotype is good in Ranchi location, but not good in other locations. Genotype 2 2671, ICPH 2671, Genotype 2, Genotype 3 2740, Genotype 1 3933, and they are the stable genotype. Genotype 9 3762, they are the stable genotype, and same result given by the Ewart Russell model. We can say our result is coming good because both are method giving the same information 2 3 1 9, and Ewart Russell model also given the genotype is stable. And this is the based on mean performance, this is the standard heterosis over the checks. Means black color is showing the Maruti, this is the checks, and red color is the Asha. This is the standard check of the pigeonpea. This is showing the standard heterosis based on first individual location, after that pooled basis. You can see the Ranchi is the variate, so much variate from the standard checks, and showing the so much highly mean performance above 2000 kg per hectare, and other locations less than 2000, and pooled basis also coming less than 2000. So we can say Ranchi is the best location for growing these hybrids. And you can see clearly in this graph, this is the standard heterosis over the checks on the pooled basis. These are the hybrids, green color hybrids, and black color in Maruti checks, and red color Asha. You can see the one line is there, that is the you can see directly, you can see what is the difference between means this is Asha and this is Maruti, and the hybrids. The you can see hybrid site and check site. What is the difference between the yield of the checks and hybrids? You can see another one, Asha. Asha is very good variety, but hybrids also varied from the Asha also. They beat also Asha variety in we can say in terms of yield. So hybrids are very good. Hybrids are very good, and you can see 3933, 2671, 2740, 4490, and 3762 are very good hybrids. They are showing very high yield compared to both the checks, and you can see the difference from the checks to hybrids yield. These are the stable hybrids for different characters. For grain yield kg per hectare, ICPH 2671, 2740, 3762, they are the best hybrid. Some color is there. 2671 is the showing stability for most of the character. You can see green color. 267 everywhere is there except secondary branch per plant, and 2740, 3933 also is good hybrids. They are showing stable for maximum yield and yield contributing characters. Finally come to conclusion. Ranchi is the most of the hybrid source high yield compared to other two locations, and Patancheru location are suitable for early maturity. This is the basic difference from Ranchi and Patancheru, and on maturity 1 month difference is there. Here in Patancheru I harvested my trial in January, but in Ranchi and other locations here I harvested my trial in February. So 1 month is difference is there. That's why I think yield variation is coming. So Ranchi, they from flowering to pod setting, the Ranchi taking more time. That's why yield variation is coming, I think. And they are the stable hybrids. ICPH 267 is stable for 5 characters. Grain yield per kg per hectare, days to 50% flowering, days to maturity, plant height, primary branches, pods per plant, seeds per pod, and simultaneously ICPH 2740, 3933, 3762 also stable for 5 characters. This is the acknowledgement. I want to thanks for Dr. KV Saxena. He gave the wonderful opportunity to staying in KSIConnect, and he accepted me as a student, and it's my privilege to I am the one of the student of Dr. KV Saxena, and I want to thanks our pigeonpea department and support the each and every aspect for me. And I want to thanks my Dr. A.N. Tikle from my college. He is my supervisor. And after that, I want to thanks Dr. Avsik Rathod and team for giving the biometrical analysis. And I want to thank special thanks to Dr. Rojana Mulla and team for LSU for accept me as a research scholar, and also I want to thanks Housing and Food Serving Committee for giving me our wonderful food and for providing us food. So thank you.
Very nice.
Yes.
I'll just
If you have any query.
thank you. Very good seminar.
Thank you, sir.
And I saw a lot of improvement between yesterday and today. Good.
Yes, sir.
Keep it up.
Thank you, sir.
Okay. So now we are ready to ask questions, and first opportunity to Dr. Anamula, and then we'll open up for scientific discussion. Anna.
Yeah, I mean, I might not be technically into this, but I've seen, all the graphs where
Yes,
the
madam.
quadrants are. And then, but then when I was looking at your conclusion, I get to understand that Ranchi is the best.
Yes, madam.
In terms of all these different hybrids. But I've seen so many variability in terms of yield, plant height, and it seems in almost all different locations you have these variabilities,
Yes.
okay, according to these different parameters. So, but then at the end you're saying Ranchi is the best.
Ranchi.
So, I mean, in what way did you come up with the conclusion that Ranchi is the best in spite of these different variabilities in terms of all the different parameters?
Madam.
Question correct? You understood the question?
Yes. Madam, I told you that Ranchi is the best location. Why Ranchi is the best location? Because mature in the Pathanchero I harvested my trial in January, and Ranchi I harvested trial in February. So one difference is there. That's why Ranchi same genotype is there in Ranchi, but that genotype is taking more time to getting pod set and other things. That's why Ranchi location varied from the other two locations.
But the main question is there because you are studying genotype environment interaction.
Yes, sir.
Why it is maturing early in Patancheru, why it is maturing 1 month later in Ranchi. That kind of analysis, that kind of answer we
Yes, sir.
expect from you, and you must, because you have seen both the locations, you have all the data, latitude, altitude, everything, irrigation, non-irrigation, so you
Yes,
should
sir.
be able to define why plants flower early in Patancheru, why late in, what is the effect of
Yes, sir.
those factors on plant growth and yield.
According to my research and my knowledge, because in Ranchi flowering there is no difference between the flowering. 2, 3, 5 days difference is there in 3 locations, but difference coming in the maturity, because in Pathancheru temperature is somewhat high, but in other 2 locations temperature is cool. That's why flowering is coming more. In pod setting they are pod taking the maturity, physiological maturity taking more time. That's why due to temperature this thing is happen. Cold temperature is there in that area. Pathancheru somewhat high temperature is there.
Any other irrigation?
Yes, sir. But in Ranchi, sir, totally there was sandy soil and totally rainfall experiment is there. They didn't provide any irrigation.
Deep soils are very deep. So
Yes.
moisture stress is not there in Ranchi.
Yes.
Okay, good. Now, first I will request our LSU members to ask questions. If the opportunity, you can take revenge from me. Okay. Not so many questions. Yes, come on. Nobody? Yeah, go ahead, go ahead. Go ahead.
Yeah, you have speak of this environment, okay, and one of which is the soil. So you have stated a while back that sandy soil or something in Ranchi is different from Patancheru, which affected also in the sort the moisture available in the soil that makes the crop maintain and sustain the good buds or flowering. Is it not? So that I think one should be considered in your study? Second, did you consider also getting off types? This is because stability also should point also the off types of these hybrids. Because we are coming up with in other locations that off types are there, and still we cannot identify which source of that is causing this. So in 2671, we have seen that more farmers are having a problem on this. So did you have, because this is one way in, because of your study, so have you come up with this analysis?
Sir, in my study I didn't see off type in Ranchi and other two and Seor location. Some plants are affected by the disease and pest, wilted and sterility mosaic, but these plant I removed from the field. But
Dr. Moyer is asking about variation within a plot. Say what happens in 2671, you get purple plant, purple flower, purple
Yes.
flower like that. So those are not real 2671. They are a little bit off type from here and there. So his question is whether off types were there or not. Then from where you got the seed, right? What kind of seed you had? That is also important. Whether you produced seed yourself by pollination or you got from lab, things like that. That kind of thing you have to explain.
I got the experimental material from ICRISAT, and they sent the same material in ICRISAT, and they sent the material in Ranchi and Seor. So there is, I think, I didn't cross that material. My supervisor, Dr. KSIConnect, given me already crossed seed to me. That's why I think I have no more idea about the seeds and other things. Sir, in my no, sir. Yes, sir.
Okay, now LSU. Everybody has to ask questions. Let's start from one end. Yes. Okay, now students are here. Today is day for revenge. Okay, because we are giving only tea, no snacks.
Yes. Yes, madam. In three different locations, I have different sowing. In yes, yes. 3 sowing dates. In first of all,
Question is, which is the best date out of 3 at different locations?
yes, sir. Sir, in first date of sowing in Pathancheru, and second after that Ranchi, after that Seor. But then lastly I got, according to my observation, 27 first in 29 July, first in 29 June, after that 11 July, and after that 16 August. But in RAK Seor is somewhat crop is not good due to late sowing and more availability of the rain at that time. So according to my observation, is early sowing is good in June or July, last week of June, and otherwise first week of July.
Any other question? Not to follow.
You see, in your parameters here, this is something to do with environment, okay, with your result. Look at your parameters. You have different latitude,
Yes, sir.
longitude, and then soil type.
Yes,
You
sir.
see, this is something that you should, yeah.
This parameter you should be able to tell why yield was high at location 1. There will be other factor also, but you have also important, for example, you have latitude 17 degree, 23 degree, 23 degree. So 17 degree is potential.
Yes.
What happens? Daylight is different, correct?
Yes, sir.
So clouding is different. The heat radiation is different. So anyway, those kind of things affect front growth. But anyway, indirectly you have touched that front growth perspective. That location.
Yes, sir.
No more questions? Samir, come on. You are going to handle more students.
Yeah, thanks Uttam, very good presentation.
Thank you, sir.
Just I want to know, in all hybrids, whether the fertility distribution was uniform or there was any gradation in that?
Sir, I didn't study of the fertility restoration. Sunil study of the fertility restoration. So Sunil will handle this aspect. I handle stability aspect.
From this table, we can say that because in a resort location, the crop was sown early compared to other locations.
Yes, sir.
So by the time of October first fortnight or second fortnight, we achieve the short days and there will be more flowering. By the time of October, because our resort location is sown earlier compared to the other location, basically there will be more vegetative growth and followed by conversion to seed. That is the right reason for which we have obtained a higher yield in the resort location compared to the others.
You show me first graph, the field graph. यार ये पहले वाला ग्राफ फर्स्ट ग्राफ
हाँ सर वही आ रहा है.
ये नहीं फर्स्ट ग्राफ ऐसे प्रोडक्टिविटी प्रोडक्शन ऑफ द कंट्री
Okay.
Yes, this one. Take on please take. Right. Now my question is to both of you. Every of you. So I can repeat my question. Now you see what is happening here. I want to draw your attention on the last slide. You do this table. Right? Now question to both of you is that why you think it is like that? What you should do to improve it as a breeder? If you are appointed breeder tomorrow, what efforts you will make to make this line different? Or you must have read lot of literature. What are the reasons it did not increase? First your name.
And in this graph, we are seeing that
Sit down, hold on.
productivity is large here, productivity remain unchanged, but area and production is changed every year. So the basic reason is that the pigeonpea is long duration crop.
My question, you deal only with the last slide.
The productivity is remain unchanged because the hybrids developed very soon. ICPS 2671 was released in 2010 in Central
Mr.
Asia.
Chaudhary sir, my question is that if you see other crops, like wheat, rice, they are also self-productive. Their graph is not like that. Okay, their graph is different. Yield is increasing, but in spite of all breeding effort in the country, big national program, why you think, put your own mind on that, tell me why yield did not increase in this crop. Give me 2 points only.
The farmers not growing pigeonpea as a commercial crop. Pigeonpea is cultivating as a for fulfillment of their family food and food for family food. So that's why we are not much input. They are not providing much input to the pigeonpea. Therefore, as compared to the other seasons and other hybrids. So that's why productivity is
So this is from farmer's point of view. What about breeder's point of view? If you don't know, say no. Don't waste time. Okay. We can tell you. He will tell you. Breeders. Okay. Come on.
Sir, according to my literature and my knowledge, pigeonpea yield is not increasing because so many biotic abiotic factors are there. If we will decrease so many disease and pest and these are biotic factor and abiotic factor also there, temperature and other things. Pigeonpea is photosensitive crop. So if we decrease the biotic and abiotic factor, we can increase the yield of the pigeon.
Okay. So your next assignment is give me in writing how you can increase pigeonpea productivity if you are appointed a breeder. Okay. You give it on Monday. So don't go home. Okay. Are you going home? Oh, okay. So this is your assignment. Only after that you will get certificate from here. Because this is very important. If you become breeder from here onward, what you are going to do? You blame farmers all the time. We don't want farmers to be blamed. As a breeder, what you will do? Any aspect you can take and let me know. So that you can guide us. Young brains are always good than old brains, so that you can tell us this is was wrong, how we can increase productivity in pigeon pea? This is a very important question. Everybody can ask you, if you go for interview, they are going to ask this question. No doubt about that. Okay, clear to you? So any other question from anybody? Yes, please, come on. Now they are getting it. Ask difficult question, okay?
You had sown in different soil, in different, you had sown your trials in different places in different soil types. Have you found any significant impact on the crops due to soil type?
Yeah. I found in Ranchi, Sior and Ikrisat. 3 locations are there, Pathancheru. And I found in Ikrisat and Ranchi, sandy soil is there. But plant height compared to from Ikrisat and Sior, Ranchi location is more plant height is there. This is the I found, I don't know what is the reason, this climatic factor and other factors, but somewhat high plant height is there from other 2 locations.
I tell you why it is happening, because it's my duty to explain any problem you are facing. See, in early growth stage, temperature is high compared to Hyderabad.
Yes.
So plant respond to temperature. All the crops in North India will grow about 2 meter, 2.5 meter, or even 3 meter like that. So this is the main factor. They grow. Biomass accumulation is much higher in north compared to here, and in pigeon pea, biomass is directly correlated with yield. More biomass, of course, considering insect control is there, right? So this is the reason you find all the time. If you go to further down, Coimbatore, Kerala, you will find very small plant.
Yes.
Those kind of things are happening because of environment. So that's where the question is, when you say genotype environment interaction, this is the genotype environment interaction. Okay, good. Yes, you. Now please use your mind.
Uttam, you used 3 locations for your material. In 3 locations, you sown in different timings.
Yes.
Whether those timing you used, those are normal sowing dates, or you only intentionally selected those dates?
Actually, this is not normal sowing date. When Ranchi and Ikrisat, they are normal sowing date. But in Sior, somewhat late due to seed availability and other things. And at that time more rain is there. That's why we can't sow seed at that time. So somewhat late. But no other reason. I want to sown the seed before, but due to reason I didn't sow the seeds properly at that time. Sior. But two are the normal sowing date.
Normal, whatever farmer follow that.
Yes,
normal.
yes.
Okay, not intentionally you selected either.
No,
to sow
not
earlier.
intentionally. It's a normal sowing date.
Okay, and what about when you are comparing location wise, we have to take care of error variance, homogeneity of error variance. You find out any deviation with regard to that error variance, because you are comparing Ikrisat, Shior and other location. When you are comparing whether they are more influence of environment, in that time we have to explain individually. We call it as the homogeneity of error variance when we are talking about stability or G into E interaction.
I think that's why we discussed that. We asking whether errors were similar in all the things, all the places or not. So full analysis before full analysis.
Yes, sir.
We test the error to find out your data is very good or not. So if you do that, humidity test, darkness test, I think we are going to see whether you can pull the data or not. Because that's the basic requirement when you do multi-location analysis. See, what is happening that you should have linear data. Because why is this happening? Because all the model based on the linear responses.
Yes.
So your data should be linear. It should fit in your model, right? So if data is not linear, that model will give you wrong information, right? That's the question. If you know answer, say no. It's okay.
I don't.
Because okay, good. Now you give the answer, please. Why we do what test? How we do it?
Butler's test must be if we find some G into interaction, if it is significant, next step is we have to see whether error variance is equal in all the location. If it is one location, it is influencing very high, means in that condition, we should consider each location separately. We have to explain each location separately.
How we do it? If data is not uniform, what you do it?
Some statistical book says we have. Okay.
Transformation.
Yes.
Transformation is the only way where you can induce linearity in your data. For example, arc sign transformation for percentage, then log transformation, because when you do very big figures, then you do log transformation, under root transformation. So purpose of transformation is to induce linearity in your data so that your analysis becomes valid. Okay. I'm talking like a teacher, my God. Okay. Any other question? Okay. Thank you very much.
Thank you, sir.
Very good.